In the human body, iron exists in two oxidation states, ferrous, feeding ferric, and ferric, each playing a distinct role in physiological processes. One, ferrous iron, Fe absorption. Fe is more readily absorbed in the small intestine, particularly in the duodenum. This is the form of iron most often found in supplements due to its higher bioavailability. Transport and storage. Once absorbed, it can be converted into Phi to be stored in proteins like ferritin or used directly for synthesis of heme, a component of hemoglobin and myoglobin. 2. Ferric iron, Phi. Transport in blood. In the bloodstream, iron is typically transported as Fe bound to transferrin, a protein that helps deliver iron to various tissues. Storage. Ferric iron is stored in the liver, spleen, and bone marrow within ferritin or hemosiderin complexes. Iron's ability to switch between these two forms is crucial for its role in oxygen transport, cellular respiration, and enzyme functions. However, excess iron can lead to toxicity, and its levels in the body are tightly regulated through absorption mechanisms. Iron from dietary sources can come in two main forms, heme iron and non-heme iron. Their absorption and bioavailability differ based on the source, animal or plant, and the state of iron they contain. One, heme iron, primarily in animal sources. Form, mostly found as ferrous iron, Fe featured within the heme complex. Sources, red meat, poultry, fish, and organ meats like liver. Absorption, heme iron is better absorbed by the human body compared to non-heme iron, around 15 to 35% absorption rate, because it is less affected by dietary inhibitors. 2. Non-heme iron, primarily in plant sources. Form, found in both ferrous, Thefe, and ferric pheatmite forms, but often in the ferric state. Sources, leafy green vegetables like spinach, legumes, nuts, seeds, and fortified cereals. Absorption. Non-heme iron has a lower absorption rate, about 2 to 20%, and its absorption is influenced by factors like the presence of vitamin C, which can reduce Fe5 to Fe and enhance absorption, or inhibitors such as phytates and polyphenols. In summary, animal sources provide iron mostly in the ferrous Fe4 form as part of heme iron, making it more readily absorbed. In contrast, plant sources contain ferric, Fe, iron that requires conversion to Fe for optimal absorption. Yes, caffeine can impair iron absorption, particularly for non-heme iron, which is the form of iron found in plant-based sources. Caffeine itself, as found in coffee, tea, and some sodas, is often consumed with meals, which can reduce the absorption of non-heme iron from those meals. However, it is not the caffeine alone that is the primary issue. It is the presence of polyphenols in tea and coffee and calcium in dairy products that can significantly inhibit iron absorption. Impact of coffee and tea. Both coffee and tea contain tannins and other polyphenols that can bind to iron in the digestive tract, reducing its absorption. Studies have shown that drinking coffee or tea with or soon after a meal can reduce non-heme iron absorption by up to 60 to 80%. Timing matters. The timing of caffeine consumption relative to meals plays a role. For example, drinking coffee or tea an hour before or after meals has a much lesser effect on iron absorption compared to drinking it during the meal. For those who need to optimize their iron absorption, e.g., people with iron deficiency, it is generally recommended to consume iron-rich foods or supplements separate from caffeinated beverages, ideally with a source of vitamin C, like citrus fruits, to enhance absorption.